Hi, everybody. This is Dr. Ed Park from Recharge Biomedical. Today, we're going to be talking about exosome science. On the graph we see on the chart, of course, we have what you know, the littlest bit, and then what you don't know in the purple. But the big Pac-Man is what you don't even know you don't know. And I think this is an important concept to keep in mind as we go through this lecture. Really just a very superficial summary of exosome science for newbies. By way of disclaimer, the use of exosomes has not been approved by the FDA for the treatment or prevention of any disease. This video is for educational purposes only. It is not an advertisement for the use of exosomes. Anecdotes are not rigorous scientific proof, although they are a form of evidence. The goals of the talk will be to discuss what are exosomes, discuss where they come from, how they're isolated, what's in them, and can they be patented by companies for approval. So just to recap, this basically is a summary of what we're talking about. Inside a cell, there's a bunch of stuff going on that we don't really understand too well. Cells can invaginate and take in substances, and they can release exosomes into the blood or surrounding environment. The machinery has been studied for many decades, but we didn't really understand what it was for. So for example, we know that the nucleus keeps the blueprints of the cell, but the nucleus also produces the mRNA or messenger RNA, which is the template for making the proteins. So this occurs in what's called the endoplasmic reticulum, where there's all these ribosomes making proteins. But then downstream of that, there was something called the Golgi apparatus, which was this thing that just makes these budding little spheres. And it's levels of complexity above that we didn't really understand, and we couldn't understand because we're not cells. We don't understand how they're programmed or how they behave. But long story short, just about 12 years ago, people started saying, hey, these exosomes maybe aren't just junk, maybe they're really crucial to the way cells operate and communicate. And indeed, that seems to be the case. So just to recap, they're 100 nanometers. They're little spheres. It's just like what the coronavirus or these Pfizer and Moderna vaccines use, same thing. A phospholipid sphere can then bind with other cells and introduce their payload. They're about 1 to 500th the diameter of a cell, so they're teeny tiny. They don't have their own machinery, their own nucleus, endoplasmic reticulum, Golgi apparatus. They don't reproduce, and they don't have their own MSC, they don't have their own major histocompatibility complex antigens in general, although we'll discuss today that that's not always the case. What distinguishes them from other membrane-bound spheres or vesicles are the bigger ones can be called microvesicles. They're not nanovesicles. We're talking 100 nanometers. Microvesicles can be, 0.1 to 1 micrometer. And then apoptotic bodies, meaning fragments of destroyed cells, just are even larger than that. So it's important to talk about the fact that we're discussing the product that's intentionally created by cells. Again, as I mentioned, the history of exosome science is only about 12 years old. There's an apocryphal story, which I don't think is literally true, but I don't think most of history is literally true, that when the people in Hispaniola saw Columbus's 3 ships, they didn't recognize them because they'd never seen that. They were invisible, which I think is apocryphal. But the point is that we knew so much about molecular biology. The discovery of the electron microscope allowed us to see small, but we can't see something we're not looking for. So just as in these toy bubbles that you buy in the supermarket, nobody was looking inside these vesicles. And typically, they were looking smaller, as in proteins, and larger, as in cells. But this entire area was completely ignored, interestingly enough. If you look at the PubMed and you search for a time for exosome articles, back in 2008 is when they started publishing. There were only about 190 studies. And only in the last, I would say, 5 or 6 years, have there been a real explosion in the articles. Last year, there were almost 3,000 articles on exosomes. And the availability to treat people has only been around for under 3 years, just about, which is when I started. The relative sizes are important. So let's spend a little time just drilling down on this slide. So the woman on the right is about 1.5 meters tall. On the left is 0.1 angstroms. Angstroms are small, Planck lengths. Those are atoms, and going up from that is molecules. And then we have lipids and proteins of various sizes, ribosome. And right at that line, that black line, is where we get the coronavirus and the exosome. That's 100 nanometers. 10 times bigger than that are your mitochondria and your bacteria around that size as well. They're a little more complex, although still, I guess, single cell. And then as we go 10 times bigger than that, so 100 to 500 times the diameter of the exosome, we get typical average cells. There's a lot of variation. So people were only looking at what they could see under the microscope, and they couldn't see. You see the blue bracket down below? I don't know if you can see my pointer, but these were too small to see. So in the lab isolation procedures, nobody was looking for these. They were throwing it away. They were only looking at the cells or the stuff that was dissolved in the serum or the fluid from blood or whatever bodily fluid. They were only looking for proteins that they already knew. So again, you can only find what you are looking for. These treasure balls indeed were not being studied at all. And kind of to the point, you don't know what you don't know. Nobody even really knows, I think, how much of biological activity is mitigated by exosomes, because we've only really studied proteins and maybe limited cell-to-cell contact. But this is a very exciting and new area of study that people are very, very unsure of. Okay, so this paper is from Peng et al., entitled Focus on Morphogenesis and yada yada. But I think it shows, even in cartoon form, the complexity of what we're talking about. So we've known since the Nobel Prize for this that there are certain areas on the surface of a cell that can invaginate and form their own spheres, these sort of clathrin-coated pits for which they won a Nobel Prize. But the opposite can occur as well. And it's well known that these MVBs, you see that in the upper left, multivesicular bodies, are these packages of packages. So it's like a big hefty bag full of FedEx boxes, and it goes to the surface. It melts with the surface and extrudes them into the environment. And that's how they send their little messages in a bottle. As we see from that MVB, inside the exosomes are called ILVs, intraluminal vesicles. That's just jargon. Once they get extruded, they're exosomes, and you see them being packaged and ready. And they come from the trans-Golgi network. And that's what I mentioned to you before, was the synthesis and packaging, mixing in with other things, some perhaps DNA, definitely proteins and lipids, and mostly RNA. And RNA is the software that will tell other cells what to express and what to not express by blocking them with microRNA. But what we don't really talk about is this whole other life of the cell to the bottom right. We have phasophores, autophagosomes. So inside the cell, there's this self-destruction, garbage recycling network. Lysosomes, we've known, are ways that the cell digests and dissolve. And so this is all going on with the benefit of this endosomal pathways, or basically the cells, both externally in commerce and internally in their own regulation, use the system of phospholipid bilayer spheres. And very interesting, in the middle there, you see that part of what we're talking about in terms of identification are tetraspanins, which we'll go into just in a little bit. So again, reminded of the Pac-Man slide, what you don't know is really infinite. And the more you know, the less you truly understand, I think. So just again, to beat a dead horse, when you say, where did the exosomes come from? Well, again, this scanning electron microscope micrograph shows beautifully an MBB. It's a big hefty bag filled with these little FedEx packages. And again, as I showed you in the previous cartoon, these are being intentionally created in a bespoke fashion for some means of communication. So you see in the cartoon that they're being extruded and they're sent out as messages in the bottom. Hopefully they'll contact the cell they need to contact and mitigate the behavior. But then you see other things. Microvesicles can be larger, and there can be MVB-like clusters. So anyway, the nomenclature, the taxonomy of this is needlessly complicated. I think important to say, we don't understand how this whole process is regulated. Above me, if you ask where do they come from, in a quite literal sense, the exosomes that we use are from mesenchymal stem cells derived from a placenta. And the picture above is something I was called to do as an obstetrician. You deliver a baby by C-section. There's a placenta you put in the bucket. So from this placenta, they derived a master cell bank. And in that cartoon next to the placenta, we see what a master cell bank is. You grow these cells, you expand them, and you create like a clone army waiting for future use. And then batch by batch, as in the case of Chimera manufacturing, month by month, they thaw out some of these Boba Fett clones, if you will, and they expand them. They let them produce their exosomes, and then they start anew next month. It's kind of like factory farming of chickens, only the chickens don't stay around longer than a month, and they're just harvesting the fresh eggs or exosomes in that analogy. When we talk about taxonomy and identification, well, there's an infinite variety of exosomes, and you could say, well, let's look at the tetraspanins. So the tetraspanins, as I mentioned, are these proteins that are embedded in the phospholipid sphere, and they're embedded by virtue of these 4 alpha helices. You see these yellow balls, 1, 2, 3, 4. That's what tetra is 4. Spanin means, I guess, to span the membrane. So they all share this morphology. And on the exterior surface, they have 2 binding regions, one shorter loop and one longer loop. And that helps with identification and presumably binding. When we talk about MSC exosomes, we're talking about 3. They're kind of multiples of 9, I guess, CD9, CD63, and CD81. And 63 is typically the most specific. But you look here, there's about 30 different tetraspanins. And of course, things like major histocompatibility complex antigens can also be considered surface identifying factors. But, you know, this is very, very complicated, and there are many different kinds of tetraspanins. I was having dinner last week with a scientist, and she mentioned she's working with neural stem cells and exosomes, and that they have other tetraspanins, which might be listed here. Of course, when you look at the literature, people who are studying exosomes in the first like 6-7 years, it was all like cancer research because that's where the money is. So they were looking at exosomes from people with cancer and saying, well, what can we intuit or what can we deduce is the phenotype or the expression, the result or the cause, they weren't really sure of cancer. And so there was a lot of research done into seeing when a system is awry, deranged, what do the exosome contents look like? Indeed, it's a chicken and egg thing. We don't really know. We do know that's scary that if you take exosomes from somebody who's getting cancer, it can help transform a non-cancerous patient's phenotype. And likewise, if someone's getting better from liver fibrosis, and you give that exosome to somebody who has liver fibrosis, they can improve. So the question is, do the bad exosomes come from the disease, or do they cause the disease, or is it not that simple? Is it both or neither? In the future, of course, perhaps we'll have exosomes from different stem cell types, people with, you know, Alzheimer's resistance or different genetic conditions that can be mitigated by exosomes. But right now, the reason we do MSC exosomes is because MSC exosomes are very special. The mesenchymal stem cell, as I talked about in, I think, lecture 24 or 5, go check it out. It talks all about the MSC. It's a special kind of cell that orchestrates. It's the quarterback. It's the barefoot doctor. It goes around and it specifically heals. It shuts down inflammation, which is super important, as inflammation can be a somewhat unwanted and untoward effect of different conditions. And then it signals regeneration. It can help oftentimes for local stem cells in their niches to recover. So at this point, when I say exosomes don't have surface antigens other than these tetrasphenins, that's what I mean. But there are exceptions. There are many different exosomes, just like there are many different kinds of songs. If you go to webinar 24, webinar 24 was quality assurance. And we talked about comparing exosome brands out there in the market. And we just discussed briefly how these things are isolated. Basically, centrifugation is important. In other words, spinning something at 1,000 Gs, I guess that's a lot of Gs. And so the gravity acts on particles of different mass differently. So it used to be that this stuff was thrown away. Now we use ultracentrifugation to isolate them. Another methodology that can be employed is these microcylinder fibers with these little hairs on them. And you can see the little hairs are just the right diameter to trap the exosomes. There are also bead agglutination kind of methodologies, but it is important to get rid of the big stuff because those are often decomposed cells, and the little junk also. What's in exosomes? Well, you can do proteomics, which is the study of the proteins. They have these commercially available assays that try to identify what proteins we're talking about. And then you have RNAomics, which is the same thing. They'll have kits, which will have the known mRNAs, and you can try and detect what's in them. But of course, it's kind of like saying, well, what notes are in that song? Well, does that really tell you anything? You can have an F sharp or, a C sharp, but the experience of music is very different to somebody who enjoys music or who understands music. When we're talking about exosome science, it's really like we're trying to study something we don't have ears to hear. We don't have the emotions to perceive. We don't have the memory or the context to give it genre. We don't have even a perception of time course to give it meter and rhythm. So our attempts to understand exosomes are like taking a snapshot or just guessing at what the movements mean without even understanding the music, if you will. So it's tough. It's tough. If I get a snapshot of something playing through my speaker, it will look like a waveform. But if I listen to it over time, and I listen to it in the context of music theory and harmony, and then in terms of melody, and in terms of genre, and in terms of my mood and my memory, that song has completely different meaning other than just noise. And the problem is that our limited understanding of what these proteins and mRNAs do in the context of cell behavior, in the context of a complicated dialogue, is very limiting. We may never understand what the heck is going on with exosomes, but people are going to try and monetize it and improve our ignorance to some extent. So on the final note, I just want to close by asking, can you patent something that's alive? If you want to go a deeper dive into this, see webinar 7 of 9 of the first 9 foundational lectures I did a couple of years ago. And it talks about the FDA and its role in approving drugs. If you look at the chart down below, what we see is that from 1980 to 2010, the explosion, or at least the increase of the number of patents for certain things, new breeds of animals, new breeds of animals in terms of genetic engineering in the blue, in terms of stem cells in the red, uh, and in terms of other things, plants in the green. So people do try and patent certain things, but is it possible? Angelina Jolie got a prophylactic mastectomy for BRCA, and she probably got a really big paycheck for that. And Myriad Genetics was trying to patent the BRCA gene. The Supreme Court struck it down and said, no, you can't patent that. But certain things like synthetic DNA, like the Pfizer vaccine, because it's genetically modified, it can be patented and owned and sold. So that's it. It's kind of a brief summary. I know a lot of people already know most of this, but I just wanted to emphasize that what we don't know, we don't even know, is not shrinking, but ever expanding as we learn more about the field of exosome science. So I think the thing that is the most mind-blowing is that up until just 12 years ago, that wasn't that long ago, no one even studied this, even though it was glaringly obvious that cells were constructed to make vesicles and they had some function. Because there was no funding for it and nobody had the dogma that this was important, all that stuff just got thrown out in all these samples. So something that could have been studied was completely ignored because you can't see what you're not looking for. But furthermore, now that we've been able to capture and see these with our limited crude tools of proteomics and RNAomics, we're trying to convince ourselves that we understand what's going on, whereas the levels of complexity of cell behavior communication through time in a dynamic environment with mixed genetics and epigenetics is really very, very complex. And so maybe this will never be through the FDA because, again, living things are dynamic systems, and can you really patent a living cell? Remains to be seen. Some of the people in this field want to immortalize and maybe think of it in terms of that way. But yeah, I think at this moment, as a clinical medicine doctor, I'm just trying to help people with the exosomes from the specific stem cell type. And we're seeing pretty good results. So next week, I'd like you to check out in case you missed it lecture. I'm going to review how I got involved with exosomes and talk about some of the many blogs, videos, and lectures that I've done on the subject. So that'll be a recap. At this point, I'd like to turn it over to questions. If anybody has a question, feel free to type into the chat or raise your hand. We have a question from John. Do the new super optical microscopes allow us to see what was previously invisible? I'm not sure what that is, but I assume that you mean some kind of microscope that's better than the old optics. I mean, if you look back, you know, they're saying they can take pictures of single atoms now, which is super groovy, cool. But yeah, we do see that the blue bracket is the light microscope. The electron microscope is the magenta bracket. There was an overlap, but certainly there are some, when I was visiting the Chimera lab, they had some cool microscopes that could see very single exosome level and radio label the CD63 on them. So yeah, we can get down to the single exosome level. But again, the devil is in the details. It's how you're going to use that info. And remember, when we talk about exosomes, we talk about, you know, the eggs coming from a batch of chickens. It's a stochastic or statistical analysis. Like every exosome is not exactly the same. So when they do every time I order, I get a composition. I know for sure they're not infected because they test for all the infectious diseases and it's the same donor and there's no introduction of disease. But if you look at some of the proteomics and RNAomics, that's a population average. That's not like every exosome is exactly the same off the line like an iPhone. No. But yeah, I think that the microscope technology is improved. Any other questions? I can unmute John.
Yeah, with regard to these microscopes, the The problem with the electron microscope is that it only allows you to slice up dead things. So you take the cell, embed it in some plastic or wax or something, and you make a micro slice of it with a microtome, and you're studying slices of dead cells or dead things. It's no use to study behavior of something like a puppy. Uh, you can't slice puppies up and look at their behavior, but optical microscopes don't kill the things that are being observed. That's why the extension of the optical microscopy realm down into these smaller size things is so crucially important, because it allows you to see the behavior of these things. And so, and I believe that there have been some things. I did hear about the thing about they can actually apparently claim to have a visible observation of an atom, which would imply they could potentially visibilize every little thing. And so I would be inclined to attend to this whole realm and get to the state of the art with regard to the optical microscopes, just to get to the cutting edge.
Yeah, no, that's great. That's a great point. That's a great point about scanning electron microscopy. You have to freeze and cut it, and yeah, it's just literally a snapshot. Thank you for sharing that. Yeah, good point. Okay, any other questions, comments? Larry asks, what's the difference of MSC treatment and exosome treatment? Well, for our intents and purposes, they're the same now. Oh, I'm sorry. You say MSC treatment. MSC treatments, if you go into Foundation Lecture 5 of 9, that's tinyurl.com, exosomes 5. I discussed the history of joint regenerative therapy. And in that, we talk about the difference between off-the-shelf stem cells versus exosomes. And I really think this is a benefit because stem cells are big and they have these histocompatibility antigens. And when you do studies, with radio labeling, we discovered that your immune system quite rightly destroys them within, days. They're not even there anymore. During that time, arguably, they could be making a ton of exosomes, but they're probably not going to be engrafting, meaning planting and growing or expanding. So the problem with MSC cells themselves is they're very expensive. You have to go overseas, and biologically, logically, and immunologically, they get destroyed quickly and may be limited in their efficacy. Having said that, some people do report benefits, so we're not really sure. But I think the quality control issues, the immune incompatibility, are just not as much of an issue, at least in my opinion, from what I've seen with exosomes. So I think exosomes are more affordable, a better value, more predictable in their use, and there's no reason why they would be rejected or ineffective. Usually, 9.5 times out of 10, the patient feels something. So, good question. Any other questions? Anybody want to ask a question or chime in? Larry asks, is a treatment center in Mexico? Yes, there are multiple treatment centers in Mexico, in Panama. And again, I think, most of them are pretty adept at doing cell husbandry, but, you never really know about the quality because it has to do with the person's ethics and their QA systems, and one batch may vary from the next. And, someone cuts corners in terms of infectious disease screening. You just don't know. I mean, it's like if you've ever worked in a kitchen, you know that a lot goes on, and maybe there's cockroaches and day-old food. You just don't even know, and it's really scary when you're talking about that in terms of people's treatment and their bodies and their health. Not to say that being in the U.S. is any safer or better, but just in general, the devil's in the details. Any other questions? Okay, so I think next week we'll do the in case you missed it lecture. We'll talk about the history of how I got into this and what I've been doing, and hopefully that'll give you a good resource list of things to review if you're interested. All right, everybody, this is Dr. Ed Park completing a webinar on exosome science. Thanks for joining, and we'll see you next time.